Good morning, everyone. So, on the behalf of the Shared Genes group, it gives me immense pleasure to warmly welcome all the respected scientists, faculty members, researchers, students, and participants to the today's lecture. At Shared Genes group, we believe that science grows stronger when knowledge is shared. We are pleased to invite you to the third lecture under the Share Genes Knowledge Lecture Series. As a part of this initiative, distinguished scientists Dr. Gyan Prakash Mishra and Dr. Anil Kumar C have previously delivered insightful lectures, and their talks are available on our YouTube channel. So today's lecture is especially important and very timely. Agriculture is now entering a new era where genetics, data science, and artificial intelligence are coming together to transform the way we breed crops and address the global challenges such as climate change, food insecurity, emerging diseases, and growing demand for the sustainable agriculture. Predictive breeding is rapidly becoming one of the most powerful approaches in the modern crop improvement, helping scientists to make faster, smarter, and more precise breeding decisions. To speak on this highly relevant topic, we are truly honored to have with us one of the world's most distinguished scientists in the field of maize genetics and plant breeding. That is Dr. B.M. Prasanna. I am sure that many of us here already know Dr. Prasanna sir for his remarkable scientific contributions and global leadership in agriculture. Nevertheless, it is truly our privilege to introduce such an eminent scientist to all the participants gathered here today. Dr. Prasanna sir currently serves as a distinguished scientist and regional director for Asia at CIMMYT, and also as the managing director of the Borlaug Institute of South Asia. An outstanding academician from the very beginning, Dr. Prasanna sir completed both his MSc and PhD in IRI in genetics from the IRI New Delhi as a gold medalist. He later served ICR IRI for nearly two decades as a scientist, teacher, mentor, and mage program leader, guiding several PhD and MSc students from the India and abroad. His scientific journey then expanded to global stage through CIMMYT, where from 2010 to 24, he served as a director of the Global Mage Program based in Nairobi, Kenya. During this period, he led multidisciplinary international teams across Africa, Asia, Latin America, focusing on the development and deployment of high-yielding, climate-resilient, stress-tolerant, and nutritionally enriched maize varieties for rain-fed and stress-prone tropical environments. His contribution greatly benefited the millions of smallholder farmers and significantly strengthened global food and nutritional security. Dr. Prasanna sir also led the CGIAR research program on maize agri-food systems and later spearheaded the CGIAR Plant Health Initiative, coordinating the several CGIAR center and international partners to address the major agricultural challenges through science-driven innovations and global collaborations. His exceptional scientific contributions have earned him numerous prestigious national and international honors and awards, including the Dr. M.S. Swaminathan Award for Leadership in Agriculture, the Shanghai Magnolia Silver Award from the Government of China, and the 2025 International Award, and recognition as one of the top agri-food pioneers by the World Food Prize Foundation. Sir, we are deeply grateful to you for accepting our invitation and taking time to interact with us today. We are eagerly looking forward to learn from your insights and experiences on this exciting frontier where genetics, AI, and agriculture converge to shape the future of crop improvement. Once again, I warmly welcome all the participants to this lecture session. And now, with immense pleasure and great respect, I invite Dr. B.M. Prasanna sir to kindly deliver his lecture. Thank you.
Thank you so much, Ravana Siddhaya and the Share Genes organizers for this Excellent opportunity. Can you please confirm that whether my screen is visible and I'm audible
Yes,
clearly?
sir, it is visible and you are
Okay.
audible, sir.
Thank you again. And I could see many of my colleagues as well as my students from IRA who are occupying significant positions in the National Agricultural Research System also there among the audience. Warm welcome to them. The topic that Jeans asked me to present is on predictive breeding and how genetics, artificial intelligence, and agriculture are coming together to transform a very important scientific area for humankind, that is breeding. To begin with, let me
Recording in progress.
let me first start with what CIMMYT is for those young colleagues who may or may not be so aware. CIMMYT is in fact International Maize and Wheat Improvement Center with its headquarters in Mexico and established way back in 1960s by this great legend Norman Borlaug, who had introduced the semi-dwarf wheat varieties from CIMMYT into India, and together with Swaminathan and a suite of colleagues at that time, sparked off the Green Revolution in South Asia. CIMMYT is a truly global organization. It is presently operating in over 80 countries, including in Latin America, Sub-Saharan Africa, and Asia. At many of these countries, we have offices, those which are indicated by these pins, while our headquarters remains in Mexico. CIMMYT has a significant presence both in Africa as well as in Asia. The Borlaug Institute for South Asia is a joint initiative by CIMMYT and the Government of India. It was established in 2011 and at three different research stations, one in Ludhiana, Punjab, another in Jabalpur in Madhya Pradesh, and the third in Pusa in Bihar. These three research stations constitute a very significant asset to both CIMMYT as well as to the national system, addressing some major challenges affecting South Asian agriculture. So with that very brief introduction about what CIMMYT and BISA are, let's now go on to the topic. Plant breeders now have to indeed deal with a diverse set of challenges. These challenges include how to stabilize and increase yields despite a changing climate, how to withstand the transboundary pests and diseases with much fewer crop inputs, especially application of chemical pesticides, how to maximize the resource use efficiency, be it with regard to water, land, and nutrients. All these have tremendous impact on the way in which we fight climate change or pests or feed growing populations, conserve water, and in the present context of conflicts across the world, how to depend less on chemical fertilizers and grow more food and more nutritive food on less arable land. So therefore, this set of challenges together with climate variability and extremes, human conflicts, economic slowdowns and downturns, and the unaffordability of healthy diets to a vast majority of resource-constrained population, all these challenges do impinge a lot on the way in which plant breeding has to be done, organized, and implemented at present and in future. Breeding strategies cannot remain constant. They do need to evolve, and not just evolve, they need to evolve much faster than ever before. For four major reasons, which I put it across as organizational, technological, structural, and process related. At the organizational level, we do need to, all the students, faculty, everyone involved in crop improvement have to be cognizant of the changes in the external world that drive the need for these changes. Disruptive innovations that need to be integrated faster, in our breeding schemes. The changes in operating practices to become much more efficient in terms of breeding than ever before, and how best to optimize and leverage our physical assets, our capabilities, and the roles that need to change. So this is depicted in a very beautiful way by some artist as this caterpillar asking this butterfly, tell me more about that 6-week plan that you have tried, and you made yourself into such a beautiful creature. That means it's a process of continuous improvement. And that process of continuous improvement has to become an integral part of our organizational strategy. The science behind breeding has also changed along with those challenges significantly over the last 2 decades, especially. High density genotyping platforms have evolved. Genomics assisted breeding is being implemented across the global north and global south, including marker assisted selection, genomic prediction, and gene editing. Phenotyping advances have come both on station and on farm. Technologies to accelerate breeding cycles like speed breeding and double haploidy are much more adopted than ever before. And coupled with all this, one area which is less recognized but which has made a tremendous impact is the way in which statistical designs and software have come. Breeding data management systems have evolved along with the breeding analytics. And more recently, these have become much more stronger by infusion of artificial intelligence, including the machine learning and deep learning models and algorithms. Let me give a brief uh, indication about how things are changing in breeding. To me, the first most important thing we must recognize is there is no longer conventional breeding and molecular breeding. Molecular breeding has become an integral part of modern crop improvement strategy. Long time back, people used to have even debates in colleges and universities whether molecular markers need to be adopted by breeders. But today, there is absolutely no denial of the fact that these have a profound influence on genetics and breeding, be it plant systems or animal systems. Look at the way in which markers have completely modulated the way plant development is analyzed, population genetics and evolution, analysis of genetic diversity, structure and function of genomes, as well as accelerated breeding and quantitative genetics. All these areas have been profoundly influenced by molecular markers. This has also happened because of the technological changes that have simultaneously happened. Today, we have a diverse genotyping and genome sequencing platforms that enable you to use molecular markers at low density or at medium density or at high density in different crop plants. Many of these platforms are available in India and being used quite routinely now than ever before. So the first thing students must understand and need to be familiar and need to be practically trained at is not only the breeding strategies for different types of crops, but also the practical knowledge of utilizing these molecular markers for various purposes. The second major development is with regard to the way in which we undertake phenotyping. Gone are those days where we have to intensively put our efforts or labor through manual phenotyping. And now we have, thanks to the developments under the sensor technology, today we have an opportunity to do high throughput phenotyping both at the field level as well as at the laboratory levels. So look at this beautiful graphic given by Frisch, Nito, and team in 2025 that talks about different types of sensors, be it RGB, hyperspectral, thermal sensor, chlorophyll fluorescence, LiDAR, for analyzing an array of traits in diverse crop plants, morphological or structural traits, physiological functional traits, and component traits of grain yield, including plant height, biomass, canopy coverage or senescence, stay green, plant maturity, diseases and pests, chlorophyll, nitrogen, water content, and even grain yield parameters. Different sensors have different kinds of applications among this. The RGB and the hyperspectral sensors, they cover a broad spectrum of traits. Thermal imaging is particularly suited for analyzing the water stress status as well as the biotic stresses. LiDAR is for structural features, and chlorophyll fluorescence has been being used for many, many years for analysis of photosynthetic activity. So, different sensors are available at a much lower cost than ever before to undertake high throughput phenotyping for a wide range of traits. I will come on to this subsequently. The next most important advance, besides molecular markers and phenotyping methodologies, are the technologies that can accelerate breeding cycles. In case of maize, we have the fortune of undertaking double haploidy. What CIMMYT did in the last 15 years is to develop tropicalized haploid inducers with high haploid induction ability. These are genetic stocks, normal genetic stocks, but with high haploid induction capacity. And these are now distributed to over 50, 60 institutions globally, including in global north and global south. More than 45 countries have benefited from CIMMYT's tropicalized haploid inducers. In addition, we established three platforms for undertaking maize double haploidy. One is at Kebako in Kenya for Africa. The other is at Kunigal near Bangalore in India. And the third one is Agua Fria in Mexico to serve Latin America. These double haploid facilities in maize generate thousands of completely homozygous lines in diverse populations, tropical populations, not just for CIMMYT, but also for the national programs and for interested seed companies. In other crops where such technology is not available, what is the option? That's where you go to speed breeding methodologies, where you, through controlled light dark schemes, you can accelerate the process of crop cycle. And line development, field testing, and trials to undertake can be accelerated through speed breeding that is conducted typically under glass houses or field houses. In case of wheat, we have such a speed breeding facility at Toluca in Mexico. And similarly, IRRI has established a similar speed breeding facility in ISARC Varanasi. The national system has also, ICIR and the institutions have also set up such facilities at different locations. These are now extremely important for deriving climate resilient varieties at a much faster pace than before. Quantitative genetics is a fundamentally important part of plant breeding. And this quantitative genetics has also been reinvented in the last 10 to 15 years, particularly. This guy is one of the best quantitative geneticists in the world, a Filipino by the name Rex Bernardo. Rex Bernardo works at University of Minnesota, and one of the single most influential quantitative geneticists I have come across, and writes wonderful articles. So those who have not read his articles, please go through it. His article published in Heredity 2020 talks about reinventing quantitative genetics for plant breeding, something old, something new, something borrowed, something blue. It's a very funny way he puts it across, but what he writes here is very profound. Over the years, quantitative genetics in plant breeding has become increasingly empirical and computational and less grounded in theory. This trend will continue as the amount and types of data available in a breeding program increases. So, what has radically changed quantitative genetics and plant breeding? First of all, again, cheap and abundant molecular markers in different plant species. The second is computer technology, the way in which we can undertake data analysis using billions of data points, simulation capacity at unprecedented levels. So, if there is one of the best simulation scientists working in plant breeding, undoubtedly it's my friend Rex Bernardo. This has also influenced the way in which artificial intelligence has come into play in the breeding programs. Today, artificial intelligence can influence multiple ways you do plant breeding or animal breeding. How to set the breeding goals, that means which traits to breed for. What are those farmer driven traits? What are those consumer driven traits where you can have maximum returns on investment? How to use artificial intelligence to collect and process data for phenotype of those target traits, including yield, nutritional quality, resource use efficiency, biotic stresses or abiotic stresses, prediction of performance of genotypes, selecting parents for crossing programs, targeting new varieties in different agroecologies. In all these areas, artificial intelligence is very, very critical. That's why in some years back, we came up with a highly cited paper in Trends in Genetics on artificial intelligence in breeding, and also published how to leverage automated machine learning for environmental data-driven genetic analysis and genomic prediction in maize hybrids. As you can see, most of my co-authors here are not only from CIMMYT, but from China. It shows the way in which China has adopted AI models in plant breeding much, much more faster than many countries. If we have to summarize where can artificial intelligence help the most in breeding strategy, this is a complex diagram, but it's, if you look at it or see it very carefully, it is one of the most beautiful graphics that was included in a publication by Fritz Netto in 2025. So these are different stages of the breeding funnel, starting with a large number of genotypes in stage 1, going to stage 2, stage 3, stage 4, stage 5, right from parental choice and crossing, generating segregating populations, undertaking early selections, then multi-environment testing of a limited number of hybrids, then narrowing these down to 2 or 3 hybrids that can go for validation in the farmers' fields, and then final recommendation and then release of cultivars. As you can see, the genetic variance varies across these different stages of breeding. As initially lower selection response, and as you go forward, it becomes higher selection response. High genetic variance to relatively low genetic variance. Fewer replications and trials to more replication trials. Lesser heritability values to higher heritability values. In all this, various data layers come into picture: pedigree data, genotypic data, environmental data or envirotyping data, as some people call it, phenotyping data, high throughput phenotyping data. All this means there are multi-omic data layers at different stages of breeding programs. Computational capacity and modeling becomes extremely important, including use of artificial intelligence, genomic selection, prediction capabilities, simulation capabilities, so that you can take selection decisions dynamically at various stages. So where exactly AI works best? AI can work very well in these 3 particular stages: parental choice and crossing, advancing segregating progenies, and making early generation or early stage selections. AI can also work best when there are complex multi-dimensional questions where conventional statistical approaches cannot capture very well the complexity involved in it. But the more complex the data set becomes, or more parameters come into the model, the bigger are the data sets that you need to create to make reliable or robust inferences. Do not think that I can use AI on a small number of 100 genotypes evaluated in one location for one crop season. That's it. If you have very good genotypic data on thousands of lines or segregating progenies, lot of environmental data, management data, coupled with this phenotypic data, then AI becomes extremely useful. So this is the first important message that I would like to give. AI is not useful if you are working at a very low scale. AI is extremely useful if you are working with large and complex data sets where routine statistical approaches or conventional statistical approaches cannot very well capture that complexity. That takes me to the question of what is predictive breeding. Predictive breeding basically is a data driven approach that leverages this genomic, phenomic, environmental data and AI based models with the aim of reducing our reliance on traditional phenotypic selection and enabling more accurate and cost effective breeding decisions. That means three particular areas where AI can be of great benefit. How to make inferences from complex multi-dimensional data sets, including this phenotypic data, high throughput phenotypic data, genotypic data, environmental data, management data. How to more efficiently implement labor intensive tasks. How to analyze the data and take decision support. So predictive breeding is absolutely critical. Gone are those days where all these thousands of genotypes need to be evaluated at all the locations and laborious manual phenotyping needs to be done. You can do these thousands of genotypes evaluated in a much more cost effective way by using statistical models, partial replication designs, as well as AI based approaches. For example, this is the work on high throughput field based phenotyping, which when I became the MAIS program director in 2011, we started working on high throughput phenotyping at one of our major breeding stations in Africa at Harare in Zimbabwe. Mainasara Zamanallah, one of my best scientists for phenotyping, he then propelled this work and then optimized canopy cover and senescence. How can we do this for our drought screening and nitrogen screening experiments at Zimbabwe? Then we extended this to how to measure ear phenotypes using digital imaging. When I was a PhD student, I used to laboriously count the number of kernels per row, ear row, number of kernel rows per year, how much are how many, what's the 1,000 or 100 kernel test weight, all that very physically. Today, using a digital imaging process, you can automate this entire thing. That's what we are doing, both on station and on farm. A high throughput method for ear phenotyping and kernel weight estimation using ear digital imaging. These kind of technologies, this type of high throughput field-based phenotyping methodologies need to be developed across different crops. Many crops, they are evolving, definitely. But we are amongst the early leaders in terms of not only developing, but validating and mainstreaming these methodologies in our breeding programs. Where can AI come in this high throughput phenotyping? One thing is to acquire large data set through these drones, these sensors, rovers, satellites. The other is how best you can rapidly process those massive data sets. That's where most institutions are limited. You can eliminate many of the manual bottlenecks associated with the data processing through four different ways. One is what we call computer vision. That means deep learning models to identify specific traits like leaf area, leaf architecture, fruit count, or, or size or disease occurrence. Advanced algorithms to detect and quantify those structures. Early stress detection or disease detection, including nutrient deficiency, drought stress, heat stress, pathogen infections. Predictive modeling, that means how to link that high throughput field-based phenotypic data to the genotypic data. Sensor integration, how best to merge the drone data with the satellite data for large-scale monitoring of areas, for example, for disease surveillance. Some of the institutions, for example, I came across this excellent platform called Wheat AI platform. It is still version 1.0, but it's an excellent platform, nevertheless, developed by South Dakota State University. And they have even published one paper recently where they are talking about analysis of wheat spikes, spikelet counts, fusarium head blight, all these kernel sizes, stomatal counts on the leaf, all this done through a particular platform. That means how to integrate the data, high throughput phenotypic data into a platform, and how to process the data using AI-based tools or deep learning models. CIMMYT is also presently leading a Google CGIAR project to develop an in-house artificial intelligence-based image processing capacity. We are also developing at the same time fair infrastructure that means easily accessible, interoperable, and data sets or infrastructure, enabling the AI models to be run on complex data sets with easy to use user interfaces. And once the drone data is collected, image processing and data analysis proceeds to generate very clear visualization output so that breeders can take quick decisions. So therefore, one is to have the data, the other is to process the data, curate the data, annotate the data, develop relevant models, validate those models, and then deploy these models for your routine use. This is one such example again from our Google CJR project, which was provided by my colleague Keith Gardner from CIMMYT in Mexico, showing how a particular block, the data collected through the drone, can go very close in terms of plot by plot level and analyze the plant phenotypes and process the data very quickly. The second major way AI can influence our breeding, predictive breeding, is the way in which our hidden treasures in the gene banks can be uncovered. There are, for example, this is our maize diversity in Mexico, captured in our gene bank at Mexico, one of the most beautiful collections of maize, 28,000 land races that are preserved in our gene bank, from the tallest challah maize to the smallest ones, to diverse shapes, sizes, colors. This is 10,000 years of evolution of maize. How can you make use of such 10,000 years of evolution for our breeding strategy? That's where we started what we call an allele mining initiative. This initiative includes AI-based tools to support faster and more cost-effective discovery and deployment of climate-adaptive alleles from world's germplasm collections. This is being implemented, led by CIMMYT, but implemented for 5 different crops: maize, cassava, sorghum, cowpea, and rice, and provides a scalable model for discovery and deployment of such alleles in our gene banks. So AI is going to revolutionize the way in which plant genetic resources are going to be managed to have better access to what we call functional genetic diversity. Not all diversity that you have stored in the gene bank has functional value. You need to discover that functional value. And we implemented a project called Seeds of Discovery in both our maize collections and wheat collections, over 120,000 wheat collections in our gene bank at Mexico, and created what we call as the molecular atlas to navigate this genetic diversity. And this atlas provides the data, tools, and resources to allow those diverse germplasm bank users to identify that diversity of functional value for specific needs of breeding from the thousands of germplasm collections, including land races and wild relatives. So you are moving from core collections to breeder panels from these genetic resources. The third way in which AI can revolutionize our breeding is in upstream breeding. That means how best we can discover genes for a variety of functions, for a variety of traits that influence our breeding programs. So this is where it can probably be very effective in analysis and use of genomic sequences and other genomic data required for upstream gene discovery, and predict their functions, then annotate those transcriptomes and construct what we call genetic networks. For those students who don't understand what is genetic network, for example, drought stress is not simply one particular pathway that influences the tolerance to drought stress. There are multiple pathways that interact in a very complex way that lead to drought tolerance in a crop like maize, for instance. That means there is a genetic network involved in it. Understanding what genes influence that network in a major way, what are the minor genes, is a physically impossible task to do if you are doing manual data analysis. That's where AI can come into picture. So the big long-term win occurs once those genetic networks for key breeding traits are very well characterized. Then using AI again, you can ask the question, what is the quickest genetic route to improve that key trait? That means, how do I design my crop breeding program? Should I do gene introgression from multiple sources, or can I simply tweak one or two major genes influencing the trait through genome editing and then make rapid improvement? Ultimately, AI coupled with genome editing or genomics assisted breeding will build those new crop idiotypes in the years to come. This is again not a hypothesis. This is a practical work that is done again by our team at CIMMYT. Machine learning based methods for predicting functional genes, the large language models for transcriptome annotations for complex traits. So remember, it's not simply plant genetic resource management, high throughput phenotyping, but also gene discovery and genetic network construction and utilization. Another area which is also gaining a lot of importance nowadays in predictive breeding is what we call genomic selection or genomic prediction. As I said, not all, if I generate through a breeding program thousands of segregating progenies or early stage genotypes, earlier we used to laboriously evaluate all these genotypes under 2 replications or 3 replications at multiple locations, incurring lot of cost and labor. But now the methodology has changed. Thanks to the capacity to genotype all these at a much lesser cost, not more than $3 or $4 per genotypic sample, you can do genotyping now at a much lesser cost than evaluating a genotype in a single row in any field research station. That means you have capacity to now understand genomic relationships amongst your progenies and cut down on the cost of your phenotyping significantly. And that is what we call genomic relationship matrix. And that genomic relationship matrix enables you to train populations, to use the trainer populations, and then develop genomic prediction models through which not all genotypes, I will evaluate only 1/3 of this segregating progeny, and I will predict the behavior of 2/3 of the genotypes based on genomic relationship matrix. How powerful this is. This is the most exciting period at which we are living in terms of breeding, thanks to all these advances and the way in which you can collect different types of data sets, which are unprecedented. So typically 4 stages are involved here: training population design, model building for prediction, prediction itself, you do that, and then select those genotypes which are having best breeding values or genomic estimated breeding values, and then again route them back into your breeding cycle. This is what we call a breeding cycle, which goes from one stage to another stage at a much faster pace than ever before. Some years back when I was a student, it used to take typically 6, 7 years or 8 years to complete a breeding cycle. What is a breeding cycle? Not when the cultivar is developed. That means you start with improved parents, you cross them, develop segregating progenies, you evaluate them, find again the best genotype, route it back again to the population improvement process. This used to take itself 6 to 7 years or more. Today we can do it in 2 to 3 years. Thanks to double haploidy, thanks to genomic prediction, you can complete this process at half the time or even 1/3 of the time than what conventional breeding used to do 15 years back. There is a beautiful publication by Escamilla and team in 2025 on genomic selection or prediction in crop breeding. Those of you who have not read it, go through this. Again, students, if you want to be at the top of this game, you have to constantly read, constantly assimilate the best papers that are coming out, and evolve your own thinking. A good breeder is one who will keep his eyes and ears open always and constantly network, understand your own strengths. What are the strengths of others? Try to understand and complement each other. That networking, that dosti karna, is fundamentally important in breeding. And it is much more important than ever before. You cannot be an expert in everything. But if you are expert in one or two of these areas and complement very well with a good team of experts, then the world is open for breeding. So genomic prediction is, again, I don't think any other crop breeding program internationally in the global south has done as much work as we did under CIMMYT maize breeding program on genomic selection and prediction, right from evolution of strategies to mainstreaming of genomic selection, continuous improvement in those strategies, leading to 30 to 40% saving of time and cost of breeding pipelines with twice increase in genetic gains under very challenging circumstances like drought prone environments in Africa or heat prone environments in Asia or disease prone environments in Latin America. So genomic prediction has become an integral part of our breeding pipelines in all the three continents. An array of papers we published way back in 2015, 16, 17, and even till date, how to do rapid cycling genomic selection in a multi-parental population, do genomic prediction across years, genetic gains with rapid cycle genomic selection, maximizing the efficiency of genomic selection in tropical populations, applying genomic selection at a very early stage of breeding pipeline, or how to combine genomic prediction with the tropical double haploid maize lines for not just drought or others, but also for a major parasitic disease called striga in Africa. No other institution, to tell very frankly, has published as many articles on genomic selection and prediction as CIMMYT did, both in maize and in wheat. But an important learning, it's a surprise, but it's an important learning. Unlike the routine statistical approaches, quantitative genetic procedures we are applying for genomic prediction, we asked a question, can AI be helpful in improving our genomic prediction accuracies? And the answer is very surprising perhaps to many people here, but the answer is improving genomic prediction per se using AI may not be a very good use case. What's the reason? As I said, genomic selection is a tool for improving breeding efficiency and accelerate genetic improvement. Our work shows that AI does not consistently outperform our basic statistical genomic selection models, but can even sometimes produce inconsistent results. We can use AI to improve prediction accuracy when we are using multi-layered data sets like genomic data, phenotypic data, metabolomic data, microbiomic data. But the return on investment is the key factor. The return on investment is only not more than 5 to 10%, but the collection of all these data set is going to cost your breeding program twice more. So we came up with the conclusion that at least with the present methodologies, it's not very ideal to use AI as a case for genomic prediction. But for all others, there is definitely much stronger return on investment. My final segment is about on-farm testing. Gone are those days where we think that only on-station testing in breeding program is adequate enough to do cultivar development and release. On-farm testing, extensive on-farm testing, is as important as on-station testing. This is where again we came up with a kind of a revolutionary model, not to treat all the on-farm data as a homogeneous data set, but classify them, each of these farm data, on the basis of farm typologies. That means if I am collecting data on 2000 farmers in Eastern Africa or Southern Africa, I don't treat all the 2000 on-farm data as single. Each of these farm, we also analyze what are their farm household characteristics, farm endowment with regard to do they have livestock, what are the maize production characteristics in that farm, how are they endowed with what kind of crop management practices are being applied, what nutrient management practices are being done. Then you classify them into typologies. That means you are combining the socio-economic data together with genotypic data, environmental data, and crop management data, and phenotypic data. So we simply are not doing, therefore, G by E by M or genotype by environment by management interaction analysis. You are doing G by E by M by S, where S is socioeconomic data. This is an extremely important evolution in our thinking about on-farm. And we published a paper last year on plants, people, and planet on know your men and women farmers, how to ensure host farmers in participatory trials that represent heterogeneity within a target population of environment. Where does AI come into picture? AI is a very powerful tool in this on-farm data collection and analysis. You can do surveys, for example, in a country like India, in multiple local languages, where there is a target population of, for example, your genotypes are being grown in Odisha or in West Bengal or in Andhra Pradesh or Telangana. That means you are in Karnataka. Then you have multiple local languages. Your AI-based surveys can very effectively collect those responses and integrate them into a standard survey approach. That means you have a greatly accelerated data collection, much larger data set, and then analyze that data and present results. That means you can do much better decision support and interpretation. So friends, this is really important to understand that AI can help in incorporating this extra critical data into your breeding decisions, farmer characteristics based on their resource limitations, gender, youth, plus farmer varietal and trait preferences, breeding evaluation data on those traits, plus genetics, plus climate data. In essence, AI can optimize the incorporation of management, environment, socio-economic data, besides genotypic and phenotypic data into breeding decisions. How exciting! This is again the most exciting thing to happen now. Thanks to the power of AI, you can do this at a much lower cost and at a much greater convenience than ever before. That's the reason why this paper of ours in Trends in Genetics on Artificial Intelligence in Breeding gives this graphic. How to organically integrate these diverse data sets through AI and machine learning to enable further refinements in our breeding efficiencies and product placement strategies. But one thing we all need to recognize is the need to democratize predictive breeding. Many institutions in the global south, unlike the global north, still have very limited capacity. First of all, they don't understand. Secondly, they don't utilize predictive breeding due to various factors. 5 major factors: lack of well-defined breeding schemes, lack of even genotyping cost affordability. Education about when to apply and how to apply predictive breeding. Lack of computational expertise with the breeding teams. And the logistical challenges related to genotyping, analysis, making decisions within a short time frame. But what we need to do now is to bring this elephant and the mouse on the same platform. It's a level playing field. How do you create such a level playing field? How do you democratize predictive breeding? The first most important to democratize predictive breeding is to at least have AI ready data sets. Not all historical data is useful in AI. Please remember that. If you put some junk into your AI algorithm, you will get some result. But that result is never going to be useful or meaningful or robust. You need to have structured, curated, harmonized way of data collection. Then you can enable ML or AI applications. Harmonize and standardize data from heterogeneous sources into uniform formats annotated with data ontologies. Have a system to integrate multi-omic or multimodal data. High quality data, this is very critical, with proper descriptions of experimental conditions, sample types, and what are the study outcomes that we are pursuing. Machine readable, not simply stored in paper. That means data set building has to become an integral part of our culture. That data set building, I am very confidently telling you, 99.9% of our institutions still don't have in the country. And the sooner we move into this understanding of data set building and providing an architecture for AI ready data sets, we will not be able to thoroughly exploit or leverage AI. What does this mean? That means you need to have institutional breeding data management system. That means a platform for effectively manage multiple data sets, not flat files. They need to be relational data spaces. Automate routine data analytics and querying process. Visualize the capacity, visualization capacity for selection decisions. What markers to use, what lines to use, what models to use, what are the prediction values on the progeny performance so that I can go for next generation. That means you need to have data-driven decision-making capacity that combines all these things. This is a beautiful graphic. I enjoy talking about it a lot. That means how to connect seed packages, germplasm, samples, ancestors, markers, haplotypes, product profiles, environments, phenotypes, sites, trials, plots, like how you connect stars in a galaxy. Otherwise, you will be simply enjoying looking at one star or one planet, but not able to relate them, connect them into galactic structures. And to connect them, you need to have an end-to-end software system. Automated capacity for genomic estimated breeding values, GCA, general combining ability or specific combining ability estimates, rich visualization of data. That's where CIMMYT and CJR system has moved into what we call enterprise breeding system and a biometrical genetics workflow. This is an open source breeding informatics software, EBS, developed for crop breeding programs of CGIRs as well as partners of CGIR like the national partners, universities in the global south. It connects, merges, builds upon our existing software. That means fragmented solutions are now integrated into one platform, single powerful tool, so that breeders can focus on how to use data to create better varieties rather than trying to source multiple software, multiple apps from different sources. So it has a core breeding management system, a service management system, and an analytical platform. If you want to get more details about what EBS is, go into this ebsproject.org website and how to use EBS user resources National partners can also access this. The terms and conditions are available in this EBS user resources URL. Go into that and enroll your institutional systems into this. Powerful way of biometrical genetics or quantitative genetics is integrated into this EBS platform. It's called BioFlow. It's a database agnostic open source pipeline which is designed to integrate different data sets, streamline decision making, enhance genetic gains, and reduce the analysis time from days to the minutes. It connects our enterprise breeding system, our breeding management system, BreedBase, Jigwa, NASA powered databases into one platform. Let me also tell you, China has moved very fast in this direction. One of the universities in which I serve as an international advisory council member is this Hujiang Agriculture University in Wuhan. This is one of the leaders in AI and big data driven breeding. They focus not only on crop breeding, AI driven platforms, but also on livestock breeding. They developed software for pig genomics, how to use AI for smarter selection. They create technologies like next generation AI algorithms, phenotype acquisition tools, and omics data analysis, even for the seed industry. Friends, some countries, some institutions have moved at a much faster pace. Taking into consideration all these developments, they have even developed this Auto GP, an intelligent breeding platform for enhancing maize genomic selection. So it's not only CGIR, but some of the national partners, the advanced breeding institutions have moved very fast. This is again, but I would like to have India to develop its own powerful platforms for use of predictive breeding. Similarly, Australia, the coffee, the Queensland Institution for Agri-Food Innovation has a specific center, a training center in predictive breeding, where they also do not only strategic research on predictive breeding or develop new technologies, but industry immersion and partnerships, and including a massive platform for PhD studentships and postdoctoral positions. So CIMMYT and BISA are presently partnering with COFFEE and the national system on how to create a platform for training hundreds of our young students in predictive breeding from India. This is going to be a game changer in terms of training the next generation of breeders. My final slide, friends, in summary, strong understanding of, irrespective of developments in various areas, students of genetics and breeding need to have a very strong understanding of germplasm in the crops you serve and how to use in breeding strategy. That's number one. There is no compromise on that. Just understanding how to use computers or leverage or use AI algorithms or models is not going to be enough. You have to be very well versed with germplasm. You have to have passion for germplasm. You need to understand what are robust phenotyping protocols, how to have excellence in accelerated breeding using data-driven decision. The future is going to be crop modeling plus multi-omic data, plus satellite data, plus farmers information. How are our institutions gearing up for connecting all these things? That's going to be critical for institutional leaders, whether it is in the universities or in the National Agricultural Research System, so that we implement AI-based breeding design and decision support. All this means that it is not a game that only one organization can play. You need to have strategic partnerships with international organizations, which have done already tremendous work in this. You have to be open, you have to be flexible enough to understand, and you have to constantly reflect where you are weak. AI is a very useful tool, very powerful tool in our analytical armory. but it will not obviate the need for well-designed experiments and modeling approaches. You cannot simply just throw any old data, rubbish data, in an AI tool and expect to get a meaningful and robust answer. In other words, AI in analytics is not a shortcut. If students think that with AI I can use shortcuts to avoid good data sets, you are mistaken. You need to have good data sets, you need to have good understanding of statistics, quantitative genetics. If you are very good at that, you can be a modern breeder who is armed with strong predictive breeding capacity. Thank you so much for this wonderful opportunity to talk to you.
Thank you.
I could see
Sir.
Balram there smiling. Balram.
Yes, sir. We will start a question and answer session. So we have some good number of questions in chat box. So I will read out by one, and you can. C. Mahadevaya has asked that, sir, what kind of population is suitable for AI? Can we use RILs or early generation segregating population? If RIL, how it will be useful breeders in selection? In early segregating generation population, it is not suitable for multi-environment trials.
See, as I said, AI is a powerful tool in, first of all, phenotyping. Whether you are using RILs, whether you are using double haploid lines, whether you are using segregating progenies, one important way that you can collect powerful data through high throughput phenotyping or imaging images, and then how to use those images in our selection decisions. Undoubtedly, AI is very useful. So it doesn't mean that one population is better suited than others. You can do it on any type of population as long as you have a robust high throughput phenotyping platform. Secondly, in early generation selection, as I mentioned before, if your population size is very limited, it's in hundreds, AI has no advantage. If you are trying to use only phenotypic data, not genotypic data, environmental data, management data, AI has no advantage. If you are using these complex multimodal data sets, multi-omic data sets, and a large population with a reasonably large data set, then you have an opportunity to use AI where conventional statistical approaches are not going to be very useful. So remember that. If your conventional approach is good enough for doing analysis on a small population, you don't need to use AI for breeding data management or breeding analysis. But if you are using complex big data sets, which you need to use, okay, in future, don't rely only upon phenotypic data. Use pedigree data, phenotypic data, genotypic data, environmental data. Have excellent environmental station, data collection stations in your fields. Utilize how temperature or how humidity, how rainfall, how other factors are influencing your product performance or your breeding trial performance. If you are using such type of complex data sets, then AI models, machine learning or deep learning models can help you to analyze that data and to visualize that data. Okay.
Okay, sir, thank you. And is Cipra Paul has asked that, sir, is it necessary we learn machine learning and DL as aspiring breeders? What will be your advice for students in genetics and plant breeding on how to learn about ML and DL and the way they are used in plant breeding effectively?
Our course curriculum needs to change. I will not even wait for masters and PhD level students to understand predictive breeding. Okay, it has to be taught right at the BSc AG third year or final year. There has to be a course on predictive breeding, on AI-based tools. Because people, even I have seen in China, people who are 17-year-old, 18-year-old guys publishing papers in Nature and Science using AI. 17-year-old, 18-year-old guys means people who have just finished intermediate. हम पीएचडी की बात कर रहे हैं या मास्टर्स की बात कर रहे हैं, इसका मतलब ऑलरेडी 25 साल 26 साल हो गया, उससे कोई फायदा नहीं है. बिकॉज यू नीड टू स्टार्ट यंग, you need to understand what are the strengths and limitations of AI based tools, whether it is in breeding or crop management. That means teachers need to be first trained. I will not start with students. There should be a training of teachers or faculty members on these tools, because 95% of them simply don't know, period. Okay, unless they know first the theory or even practice of it, how can students know? Okay, so therefore I will start at the university level of the training of breeding community or faculty on predictive breeding. I don't know how many teachers are participating in today's webinar. Okay, but I could see two of my own students, Balram Marathi there, Niral there. Okay, so I'm glad that at least some are there. I am sure the Lal Ahmad also must be there from Bapatla. So we need to do a massive initiative. Those who are in responsible leadership positions need to please convince your vice chancellors. If you don't start now, you will lag behind, not just 10 years, 15 years, even by 30 years. Okay, so start there, teach at a very young age, advance your teaching level or proficiency as you move into masters and PhD. Make sure that once you are in a breeding work, at least the student, they just don't do any routine PhD work. Each student must have the capacity to at least understand how different types of data sets enrich breeding decisions. Gone are those days when breeding papers or things are published only by using diallele analysis or line into tested analysis. इससे काम नहीं चलेगा. So please understand that we need to go multifold in the level and capacity of our breeding work.
Sir, Bhaskar has asked that, can we integrate high throughput phenotyping to
another student of mine joined, Raja Raghupati. Good. Okay. Okay, they are all nice, very accomplished scientists, all of them. I am very proud of all these colleagues who are joining. That shows their interest, that shows their keenness to understand and to influence things. And Prasanna kya influence karega? They're very limited, but these people are capable of influencing a lot. Yes, next, go ahead.
Sir, Bhaskar has asked that, can we integrate high throughput phenotyping to speed breeding? If yes, what are the best sensors to use in those artificial LED conditions?
I would not say speed breeding and high throughput phenotyping are perfectly integratable, because the purpose of speed breeding is rapid generation advancement. That means what you develop and what it takes 3 to 4 years, you are now trying to do in 11 months or 12 months. Okay, that means you are able to generate the new lines at a much faster time in large numbers. But once large numbers are developed of those, then data collection has to be, depending upon the trait. Some data may be collected in the laboratory for nutritional quality traits, for example. Some data may be collected in the field. Most of the data, breeding data, is collected in the field. For abiotic stresses or biotic stresses, for climate resilience, without field data collection, there is no breeding. Okay, and once you go into field with such a large number of progenies, then your capacity to select accurately becomes important. That's where and the capacity to collect decisions at real time becomes important. And that's where high throughput phenotyping comes into picture. And that's where AI is a tool to analyze this data in a much faster way rather than conventional analytical approaches. So these two are two different things. Speed breeding or double haploidy to generate your segregating progenies or new lines or homozygous lines in a very rapid manner. And the second is large scale evaluation of those progeny. The more the number or the selection intensity is better. If I am analyzing DH lines, I won't analyze 100 DH lines, 200 DH lines. In each of my breeding programs, several thousand DH lines are analyzed or evaluated at multiple locations. That means lots of data has to be generated. So remember, scale, selection intensity are very critical.
Yes, sir. Ayub Deb questioning that will integration of AI models in breeding program reduce the impact of breeders in decision making, or a bit of human touch will still be required to make things easier
Very
in the
good
long term?
question. Never ever think that breeders' role is diminishing here. Okay, breeders' touch, breeders' understanding, breeders' evaluation of genotypes is absolutely critical. No matter what technological advancements happen, breeding is still is heavily involving human touch. As I told you, if I don't know what germplasm to use, okay, in my selection process, in my crossing process, if I don't understand my germplasm breeding history, where it works, how it works, what traits it will have, you will not be a good breeder. So therefore, don't ever think that technological advancements, whether it's in sensors or statistics or AI, will obviate the need for breeding or breeder. Breeder is still absolutely critical. That human touch is absolutely critical. So nobody can replace, no matter why, as long as humankind lives, breeders will have to work.
Yes, sir. Saru posed a question that, sir, are there any particular candidate genes or genomic regions in the maize that you are interested in for allele mining?
Oh, of course, there are plenty of such genes available for diverse traits. We are interested, for example, in biotic stress resistance. What specific genes or genomic regions influence resistance to major diseases or insect pests? For example, our team, I can see Suresh there, my colleague, former colleague in CIMMYT, maize pathologist. We did classic work on maize lethal necrosis, identifying first of all QTLs, then going to the gene level, identifying what genes influence resistance to this complex viral disease, then doing genome editing, and then testing them in the field and proving their efficacy. So absolutely important to understand at the gene level, but you have to go very systematically. A number of traits, whether it is biotic stress resistance, abiotic stress tolerance. Unfortunately, in a crop like maize, you cannot make major transformative changes by just tweaking one gene for drought tolerance. ऐसा नहीं होता, वो होगा भी नहीं. But for diseases, it's a different matter. For nutritional quality traits, it's a different matter. Okay, and similarly, nowadays for nitrogen use efficiency, there are some major genes or genomic regions identified, which can reduce our dependence on fertilizers by manipulating those genes. For example, in wheat, we are working on a system called biological nitrification inhibition, which can be a nitrate, which can reduce our dependence on nitrogen fertilizer by 25%. It's going to be a major breakthrough. So therefore, breeders, irrespective whether it's maize or wheat or any crop plant, there are always, you need to be interested in what developments are happening at the functional genomics level. And but not that every gene that is detected is going to be useful. That is going not. That's also you have to understand. That's why AI can help you in predicting what genes or what genetic route you must take for that particular trait improvement.
Yes, sir. Sir, hope if you have time, we will take some
Yeah,
more
yeah, sure. Go
questions.
ahead,
Yes, sir.
Raja. Go ahead.
Yes.
Yeah, you are muted. You are muted.
Just a minute, sir. I will just unmute you.
Okay, go ahead.
Yes, sir. As usual, it's an inspiring talk, great talk. I thought of asking this question from your wisdom, sir, you taught us to think very critically. My question is two things again. One, the conventional breeding, when it is spread over 12 years, indirectly we are selecting for climate adaptation. And when we go for speed breeding, we are removing that component. And the second thing, we are exposing that to high, longer length of light. We know short day, long day plants. And 22 hours of light exposure, you taught us the gene expression. Is it not a stress on the plant in the early generation? And when we use different germplasm, each has been adapted to different part of the world, and how they will impact. Is it a good strategy from pure gene expression standpoint or gene interaction standpoint?
Okay,
For instance,
let
carbon
me
dioxide.
very quickly answer that question. First of all, speed breeding is a component of population development, population improvement. Speed breeding is not about product development and performance testing. Speed breeding means what we are trying to do here is what takes 3 years, 4 years to develop homozygous lines in large numbers, you are able to do it in 11 months through a double haploidy or through a speed breeding. Exposure to an LED light and dark cycle is not a stress. But what it does is it activates certain pathways to accelerate your flowering process and thereby your capacity to generate new populations. So don't confuse speed breeding with climate resilience. Speed breeding is only for population improvement and our population development. As regards your question, by shortening, what takes, for example, in CIMMYT now from breeding conceptualization or crossing to finally releasing an advanced hybrid to a partner, it takes not more than 6 to 7 years. It used to take 15 years some time back. Now it takes maximum 6 to 7 years. Does it mean that we are compromising on climate resilience? Absolutely not. That's the reason why I am talking about not only on-station testing for managed stresses like drought stress or heat stress or waterlogging stress, which provide an excellent understanding of the genetic capability of a genotype. That managed on-station testing for a disease or for abiotic stress is absolutely critical for a product pipeline. What does on-farm testing do? Then it validates the performance of your on-station one under real farm conditions. Okay, and that wider diversity of on-farm trials will give you the confidence that what is working in the on-station when these stresses come as compounding factors. Because in the farm there is nothing like only drought or only disease or only something else. There could be X plus Y plus Z, or there can be only X plus Z or Y plus Z. You record all the data, you analyze the patterns, then you understand what kind of climate resilient traits are not only expressing strongly in a particular on station but also on farm in diverse countries. For example, CIMMYT does this on farm trials and on station trials not just in one single country like in Kenya. We do it in Kenya, Uganda, Tanzania, and other countries like in Southern Africa, Zambia, Zimbabwe, South Africa, or Malawi, or Mozambique. As many as 9 different countries we undertake on-farm trials. That means robustness of data set, broad spectrum adaptation of your genotypes. You understand this? So if
Yes,
you are
sir.
talking
Yes, sir.
about India, you don't have the opportunity to do it in multiple countries, but multiple states that are important for a product profile in rice or in wheat or in this thing, you have to do that testing.
Yes.
Unfortunately, on-farm testing is presently not an integral part of our breeding strategy. I told this to some of our colleagues in the ICR system too. We need to make on-farm testing also an important strategy. Then you will be realizing how your products are performing not just on station, how are they adapting to the diverse abiotic and biotic stresses in real term, in the real time. Okay, got it, Raja?
Yes, sir. Yes, sir. Thank you, sir. And for just for information, sir, we have done that stress memory in over a light exposure. We found a lot of microRNA. We were unable to publish it. So I request
You see,
somebody should do this.
stresses will happen,
Yes,
not
sir.
just during a double haploid induction process or colchicine treatment. Stress may happen even subsequently in multiple stages, but that should not deter you from selecting genotypes and analyzing their adaptive capacity.
Yes. Yeah.
Because
Thank you,
plants
sir.
are
I mean,
also smart and
yes, sir. Not
animals
like
are
that.
also smart. They
Yeah.
adapt.
I mean, you talked about transgenerational epigenetic memory. My request is the young generation, next generation should do research on those things instead of thing.
Yeah.
This should go, but simultaneously on the genetic side. So yeah. Thank you, sir. It's a great talk. Thank you.
Yeah.
Yes, sir. Thank you. Sir, Deshraj is asking at what cross-validation scenarios are proving to be the most reliable when predicting the performance of the completely untested populations in the new environments?
Exactly. So that's why mainstreaming genomic prediction requires cross-validation. That means what is the correlation between the data that you are getting from the genomic estimated breeding value, GEBV, versus real performance. So we did this kind of our papers, if you go through on genomic prediction, optimizing genomic prediction in tropical maize populations, they publish the data. They show what is the level of correlation. This is what we call prediction accuracy. That prediction accuracy comes through cross-validation. If your prediction accuracy is very low, there is no point going for genomic prediction. If your prediction accuracy is at least 0.6 or above, then you can very safely take genomic prediction.
Okay, so thank you. Hariam
Balram has raised his hand. Balram?
Yes, okay. I will unmute. Yes.
Yeah, sir, good morning. Thank you very much for taking time and giving the lecture to all the faculty and students of this country. We know how good teacher you are. We are fortunate to have you as our teacher, and that changed our lives. That's where I am today. And I would like to acknowledge you and thank you very much for instilling such a knowledge and confidence in us so that we could able to deliver the things. We rightly understanding what you are proposing. Actually, you are saying that national system is not having this field testing component in the farmers field and all. I could clearly see that as a very good example why nationally identified varieties are not performing the farmers field, because when you see the universities varieties are performing better when compared to that, because the release system itself integrates the farmers field testing, mini testing. Exactly this is what we are having advantage, and we are getting the data, talking to the farmers, getting the environment, information and all. As you rightly said, we are having a lot of data, like getting the farmers information, everything. But thing is, it is haphazardly collected. As you said, the data set may not fit into the AI models, everything, because we do not have the environmental data. We do not have the exactly what is required to analyze the data critically. But we need to raise ourselves to the next coming type of prediction models. Exactly saying that one. Fortunately, we got the opportunity to work in whatever you have said recently with the help of University of Tokyo. So we got one international project wherein we worked in the both rice and maize on both high throughput plant phenotyping in the field, because plant phenotyping in the lab may not serve the purpose for the breeding and all. To understand the molecular level, that is fine. But thing is to understand the actual performance in the field, high throughput plant phenotyping in the field, whatever the sensors you are told, we tried with all these cameras, RGB cameras, multispectral cameras, thermal cameras, and all these red edge camera. So around 5-6 type of cameras were used, drones, and that is also for breeding purpose. Imagine that in the 2018, 20, and 22 until 22, we worked on analyzing different data sets of this germplasm lines, try to integrate this genomic data, phenotypic data, everything into that one. You rightly said. But
wonderful. I think your university is providing the path. I'm sure that many other universities need to think about it on how to create now platforms. That's why I said the strategic partnerships are very critical with institutions that have that kind of capacity and willingness to share their knowledge and tools. And that's how you can build the next generation.
Yes, sir. Step 5, we would like to go ahead with this one. First thing is that data is very much important, and standardization of data, protocols, everything. We would like to use your help in building that database.
Absolutely.
ACS level.
जरूर आएंगे आप बुलाओ हैदराबाद आएंगे हम.
You sir, this is I think already we are meeting over online only. Already I invited you for some lectures for this Hyderabad Genetics and Plant Breeding Society and all. You delivered good lectures to the students, to the faculty and all. Thank you very much. We are in the same line, sir. Certainly we'll take your help in building the system.
Anytime. Thank you, Balram.
Yes, sir. Thank you. Sir Aryaman is asking what are some tools or techniques your team has used to increase the DH production efficiency?
So in case of DH, we have published a paper. For example, you can see these protocols are published in Theoretical and Applied Genetics in 2022. There is also a manual on maize double haploid production that CIMMYT has published. You both are accessible online. You can type CIMMYT maize double haploid production manual or the paper in Theoretical and Applied Genetics. It provides a comprehensive understanding of how we derived tropicalized haploid inducers. Earlier, before CIMMYT, these such tropicalized haploid inducers were not available. Only temperate haploid inducers were available. So we are the first to come up with publicly available tropicalized haploid inducers. So a lot of improvements have happened over the last 15 years, and still we are making improvements. We have come up with first generation haploid inducers, second generation haploid inducers, and now very soon we'll come up with third generation haploid inducers. So continuous improvement will happen.
Yes. Anirudh posed a question that why there has not been any next revolution in the last 50 years in spite of boom in molecular breeding, genetic engineering. What would be the reason?
I won't say there is no what we call breakthrough like what you have seen in Green Revolution time, suddenly catapulting yields in a massive way, because the baseline was very weak. And when the baseline was very weak, and when India achieved self-sufficiency through this dwarf wheat varieties, it was a massive movement, undoubtedly. But as I said, now what we are seeing are disruptive innovations that have come time to time. As I already said, molecular breeding has now become an integral part. Today, not even a single maize hybrid or a wheat variety is developed by CIMMYT without the use of molecular tools. Not even a single variety for the last 15 to 20 years. Is that not a revolution of markers? That means, and as I said, from 15 years, if you are able to get an improved hybrid in 6 to 7 years, almost reducing by half, is that not a revolution? So the number of climate resilient maize hybrids, today the drought tolerant maize hybrids developed by my team at CIMMYT occupies 9 million hectares in sub-Saharan Africa. When I joined CIMMYT in 2010, it was 0.5 million hectares. From 0.5 million hectares in 2024-25, it has reached 9 million hectares through massive public-private partnerships. Over 100 such hybrids released across 20 to 25 different countries in Africa. So this is a revolution. That's why very recently CIMMYT received the CIMMYT prize from Kuwait government for drought-tolerant maize work. Okay, so don't think that the only thing is we don't have personalities like Borla anymore, or personalities like Swaminathan. Okay, that is the different aspect. But it doesn't mean that we have not made a transformation in crop breeding. In multiple crops, not just CIMMYT I'm talking about, in many other institutions, changes have happened. But where are changes required? Pulses and oil seeds, for example. That's where there has to be a massive transformation. We need to change the yield levels of pulses, yield levels of oil seeds drastically if you have to meet the domestic demand and reduce the exchequer on our country. So there are some weak spots, there are some bright sparks in our crop breeding programs. Both are there: horticulture, vegetable crops, field crops, a number of crops, there are bright sparks too. So it's a mixed bag. Don't ever think that we have not sparked any major revolution in many crops. No. But remember, it's not only because of genetic modification or genome editing. A lot of silent work has happened through breeding, whether it is through conventional breeding or marker-assisted breeding. A lot of silent revolution has happened. Okay, so let's not underestimate that.
Thank you, sir. Vishwanath asked two questions, sir. First one is, given that high selection pressure on a primary trait often lead to the accidental loss of superior background genetics, what AI-driven approaches can we implement to mine our historical data and salvage these hidden and high potential genotypes?
Very, very good question. बहुत बढ़िया question है. That's where I talked about allele mining in plant genetic resource management. That's where I think, irrespective of crops, if you have an opportunity to collect good genomic sequence data, if you have an opportunity to collect good phenotypic data on your plant genetic resources, then there is a way to relate that genotypic data to phenotypic data using AI models and identify what climate adaptive alleles or what novel alleles are still there in our genetic resources that are not exploited by plant breeders. Okay, and then you design your pre-breeding strategy where you mobilize or introgress those unique alleles or novel alleles that are favorable haplotypes, not just alleles, even haplotypes, into your cultivated germplasm. And that requires massive analysis of genomic sequences of your phenotypic data, genomic sequences, environmental data at which these genetic resources have been evaluated, and then use AI models to mine that data. Okay, so that's where PGR has a very important plant genetic resources management will be transformed through AI in the coming years. We need to be ready for that.
Okay, sir. Thank you, sir. The second question is, to build effective AI models for breeding, high quality data is essential. How do you see the role of the breeder evolving in terms of providing ground truth data and validating these AI predictions?
Okay, again, as I said before, breeding is a teamwork. Okay, it's not that every data relevant for a breeding strategy is generated only by a breeder. You have entomologists, you have pathologists, you have nematologists, you have soil scientists, you have agronomists. All need to have high quality data collected and integrated into your breeding data management system. That's why if you look at our CIMMYT, we have invested significantly on data science. data science is a very critical unit in CIMMYT right now. Protection of that data from piracy, organizing that data and mining that data through highly professional quantitative geneticists and bioinformatics specialists is absolutely critical. So therefore, please don't think that breeding means only breeders will collect ground truth data. Breeding is a complex game now. If Prime Minister asks you to develop varieties, crop varieties that have reduced dependency on chemical fertilizer by 25 to 30%, can you do it alone without agronomists or soil scientists or even people who have biofertilizer capacity or microbiological expertise? You cannot. So presently I am conducting at our BISA research station a balanced fertilization experiment in rice crop. Okay, so imagine, so please do understand, breeding for cropping systems is going to be very important, not just breeding for the sake of one single crop. That means you are trying to see what will be the duration of my variety. How does it fit into a cropping system? What would be the impact of this crop on the subsequent crop? So, and how do I enhance the profitability of a cropping system, like a maize, potato, or a mung bean cropping system, or a sugarcane rice cropping system? So that means you are thinking beyond a particular crop into a cropping system. If you want to do that, then you have to think beyond simply traditional breeding. You have to work with a multidisciplinary team to collect relevant data.
Okay, sir. Thank you. Nihanika Goa has asked that any advancement, particularly for AI-driven QTL search and validation.
Very good. AI-driven QTL selection validation. Again, if you are working on a particular trait in a crop plant, unless and until this is a completely new trait where nobody has worked before, okay, you should be able to analyze what metadata exists for that particular trait in terms of QTLs. Earlier also we used to do, long time back, 20 years back, we used to do metadata analysis for QTLs. Which QTLs co-localize? What are their potential candidate genes which influence, which these QTL regions are influencing? How to narrow your QTL region, because one QTL region could harbor even 30 genes, 40 genes. It's not simply one gene. And that means you need to go from a rough map into a fine map. Identify what genes, what particular gene or genomic region is influencing that trait. In all this kind of an analysis, which requires intensive computational processes, AI can be very powerful. So that's why in predictive breeding, impinges on all these things, right from QTL mapping to QTL validation to co-localization of QTLs, identifying functional and structural candidate genes, seeing what particular candidate genes could be of potential value for a trait, and how to narrow down this interval.
Okay, sir. Thank you. Mahesh has asked that as a young plant breeders, what skills should we develop to effectively integrate genetics, genomics, and AI in future crop improvement programs?
Very good question. So again, first of all, you need to have a good theoretical exposure about the strengths and limitations of various approaches. Don't ever think that AI is foolproof. Okay, AI can give a lot of rubbish results, and we simply sometimes copy and put it in our papers or assignments or even publications or presentations. Don't ever do that. Human intelligence is always very, very, very important, as somebody asked it. You need to combine your intelligence with the artificial intelligence. You need to understand that what are the limitations of your own data set? What question am I asking here? If you are asking a wrong question, you will get a wrong result. Okay, if you ask a right question with the right data set, you will get the right and meaningful answer. So therefore, this discerning the capacity of your intelligence versus artificial intelligence, that's where a guru comes. गुरु का रोल वही है, ठीक है. ट्रेडिशनल टाइम से लेकर अभी तक, व्हाट इज गुरु's रोल? Earlier they used to teach on something else, they used to teach on blackboard or whatever, or on whiteboard, now through presentations or whatever. But a guru's role is discerning the grain from the chaff, making you understand what is valuable from and from junk, how to develop your capacity to analyze, capacity to draw inferences, putting on your head a bucket of water. As a young PhD student, you go there, sir, आज मैंने दुनिया को बदल दिया, ये डेटा आ गया मेरे पास. अच्छा, my guru also used to put a bucket of water on my head, okay, and used to tell, अच्छा प्रसन्ना, ये अच्छा लग रहा है लेकिन इसमें शायद दम नहीं है, बोलना पड़ेगा. And क्यों दम नहीं है? Okay, that's why guru is important. If you don't have a guru who can tell you what is right from what is wrong, how to put a bucket of water on your head, then you don't have a guru. Okay, so this is what you need to inculcate. Understand these questions, temper different then you are forced to think about different hypotheses. Then you are forced. That's why I told very clearly today that AI is not for genomic selection, because I gave one single word there, return on investment. Anything that you do need to have a return on investment. Is this worthy enough of my investment or my institutional investment? Will the cost of undertaking be much more than the gain that I get? Is there a more simpler solution than this? How will you know that? That's why guru is important.
Okay, sir. Thank you. Anwar is asking that in CIMMYT's genomic selection pipeline, are GUI-based tools are being used for prediction? Also, which prediction models have shown the best accuracy across breeding populations?
The best models depend upon different situations and crops. There is no single algorithm or single model that works across multiple situations. Therefore, there are multiple neural network based models, Bayesian models, there are multiple kernel truth models. We experimented on multiple types of deep learning models, and there are models that fit very well for improving selection intensity. There are models that very well fit for selection accuracy. There are models that fit very well for wheat breeding versus maize breeding. So that's why you need to work on different training populations and then experiment on different models and come up with that particular model which works best for a particular type of crop, particular population, particular trait of interest, and giving high levels of prediction accuracies. So there is no single one. So that's why it's important to go through those papers. Yeah.
Yes. Thank you, sir. Bidwan is asking that, sir, when we use controlled conditions of photoperiod, temperature, etc., to advance the generation rapidly, can we still use the phrase speed breeding if no genetic gain has been achieved?
Again, you are making a mistake. Speed breeding is not about genetic gain. Genetic gain is basically a concept where you test how well the performance of my next generation is doing compared to the original founder or original population. That means in terms of a trait, how does the trait improve versus what I started with in the beginning? That is a genetic gain, right? Whereas speed breeding is simply a process to develop thousands of lines, a population. And the population needs to be subjected to evaluation. Then you can compare that in the evaluated population, this is the mean of the trait, and in the original population, that is the mean of the trait, and this is how they compare. So speed breeding is not not directly. Speed breeding is a means to improve genetic gain. Why is it a means to improve genetic gain? Because remember the breeder's equation. The breeder's equation, the numerator has selection intensity, accuracy, genetic variance, and the denominator has cycle time. The cycle time, if it is very less, then your genetic gain will increase. If your cycle time is 4 years compared to 1 year, then the cycle time is going to be smaller because your denominator is more. You understand that? So speed breeding or double haploidy is a tool to increase your genetic gain by reducing your cycle time. Okay?
So we have still 4 to 5 questions.
Okay,
Should
let's finish 4 to 5 questions. I need to also go.
Yes, sir. Considering that CIMMYT is leveraging predictive breeding to develop the superior maize hybrid, is there any ongoing interest in developing apomictic versions of these hybrid to overcome the bottleneck of repeated hybrid seed production while retaining the hybrid vigor?
Very good question. CIMMYT worked on apomixis in maize long time back in 1990s. There was a French team which worked at CIMMYT Mexico to explore apomixis in Tripsacum dactyloides, which is a wild relative of maize, and try to introgress that apomixis from Tripsacum into cultivated maize. Unfortunately, that strategy did not work. So therefore, we do not have apomictic maize anywhere in the world right now. Whereas in apomixis in rice, there have been some significant developments, especially at University of California, Davis, Venkateshan Sundaresan and his team. You can type in your Google and get the details about Venkateshan Sundaresan or Venkat Sundaresan and his work on apomictic rice. So that is being projected as very promising. So if all things work, apomictic rice could be a reality soon. But in other crops, other field crops where grain is the most important economic produce, like in maize or in wheat, apomixis is still not a reality.
Okay, thank you, sir. How to balance between short-term genetic gain and long-term maintenance of genetic diversity in genomic selection?
Fantastic question. Somebody has asked a superb question. That's why you need to be very, very conscious about what germplasm are you using. Monitor the genetic diversity in your finished lines, in finished hybrids. Okay, and that's where it's very important. That's why human intelligence matters. If you are focused, because genetic gain will obviously increase very strongly if you are working with the closed breeding populations. Okay, the more closed your breeding population are and more cycles of selection that you impose, better will be your genetic gain. But in the process, you will also lose a lot of valuable alleles and valuable diversity, functional diversity. That's why it is important to understand what functional diversity is there in your germplasm collection and in your cultivated or released wheat genetic pool. And how do you constantly enrich this while you are trying to increase genetic gains? That's why at CIMMYT, we also do at least 20 to 25% of our pipelines. We also try to introgress novel alleles from temperate maize populations in our tropical populations. This is what we call temperate tropical introgression. So that new alleles, new diversity from temperate sources are enriched in our tropical germplasm. Similarly, in temperate regions, in like for example in China or in US, they also utilize the tropical sources to enrich their germplasm. That is tropical temperate introgression. So that process of introgression can be optimized if you know the haplotype diversity that you have. And AI can help in that. It can help you in understanding, mobilizing what haplotypes can enrich your diversity versus what haplotypes are already there and may not be so useful to diversify. You got it? So a targeted introgression strategy.
Would early genotyping of AI selected parents confirm their reliability for use in varietal development?
No problem at all. Early selections is as I said, selection is one thing, but what happens during the course of evaluation is another thing. That means how robust is your on-station testing strategy? How robust is your on-farm testing strategy? How are you taking selection decisions objectively, not on the whims and fancies of one breeder? Okay, in CIMMYT, what we do, breeder will not have a complete say on what genotypes should go finally to our announcement. There is a committee, there is a complete process, including our partners who contributed data. They sit together and we share the entire data to them and they rank the genotypes. These genotypes are very good. These genotypes are not good. These have worked very well in my country. These have not worked in my country. So it's a collective decision making process based on strengths and demerits of each hybrid. So therefore, it is not just a selection at an early stage that influences a cultivar development process. It has to be rigorous evaluation, analysis of data, and decision making on product advancement.
How to decide test cross panel to generate test cross data in hybrid breeding to calculate GCA and SCA in genomic selection?
Yeah, baat hai. Another very good question. When you select a tester for your test crossing, if you understand what is the molecular structure of your populations, okay, like for example, heterotic groups, I have A and B in maize. I know how is my A structured, how is my B structured in terms of genomic data, and where are the testers falling in the A group and in the B group. You got it? So when I choose a tester, I choose not only on the basis of their combining ability, but also on the basis of their genetic relatedness to a subgroup within my heterotic group. Let's say this is one big heterotic group. There could be one cluster of genotypes here, another cluster here, another cluster here, but they are all under one heterotic group. If I choose a tester, I will try to select a tester that is representative of each of these subgroups. How will I know? Not by phenotypic data, not by test crossing data, but by understanding their genomic relatedness. So getting genomic related, that means all the lines that I use in my heterotic group, I need to have an excellent, at least a mid-density to high-density genotypic data along with the testers. Abner Chin has raised his or her hand.
Yes, I am going to unmute them. Yes, Abner Chin, you can unmute yourself.
Unless and until Abner wants us to stop.
Yes, sir, he is not. One last question from YouTube live, sir. How predictive breeding would we explore for developing nutrient efficient crops where many physiological attributes or response or nutrient starvation guided metabolic adaptation came into picture?
Wow, very tough question. Okay, as I told you, improving nutrient use efficiency is one of the major targets for breeding community now. We need to now initiate programs on not just any genotype that can take a lot of fertilizers or dependent on chemical biostimulants or others. What exactly, how to make your crop produce as much, if not more, using lesser synthetic resources. So nutrient use efficiency is going to be a very, very important component. Not many institutions have well-designed programs for that. So that means you need to have a look at your germplasm, look at what genotypes can serve that kind of objective better, and design experiments together with agronomists and soil scientists. How to evaluate those genotypes, how to take soil samples, analyze them beforehand, the nutrient status in those soils. That means establish baselines before every crop season and dedicated blocks for analyzing those populations. For example, if I am working on nitrogen use efficiency, can I create an excellent block with the nitrogen starved plots? And where your genotype, in Africa we have such N depleted blocks for evaluating our nitrogen use efficiency in maize, where dedicated experimental blocks are used in various locations in Africa. I haven't seen such a kind of experiments done here. So we may need to do that besides manipulating, without creating such dedicated blocks, how to dynamically collaborate with agronomists and soil scientists to work on this. So this is an area which is, to me, in my opinion, a lot of emphasis will be placed in the future, and we need to work on it. Abner, can you mute it now, or you still can't mute?
Yeah, I am actually asked to unmute him, sir.
So that
now you can try.
Unmute. Okay. Okay.
You can unmute yourself now.
Okay. Otherwise,
yes, sir. It was more than like a question and answer session. Rather, it was like an interview kind of session. It took a very.
Thank God you guys interviewed me after such a long time.
Yes, sir. Actually, your expertise has guided many participants here.
By the way, sometime in August, September, October, we will be organizing 2 webinars. One, a national level webinar on AI-driven crop breeding.
Okay.
And another is a regional webinar across South Asia on AI-driven crop breeding. That would include colleagues from Bangladesh, Sri Lanka, Nepal, and Philippines, and so on. But the first one will be a dedicated webinar for national partners, national institutions. So I will share that information as it evolves.
Yes,
and this is through a project funded by the U.S. government on future-proofing Indo-Pacific agri-food security. So I am leading that AI project and that investment by the U.S. government.
That's nice, sir. We'll share it in our group as well. Yes, sir. We'll come to end this session. At this moment, I would like to share a small but meaningful connection behind this today's lecture. I had opportunity to briefly interact with Dr. Prasanna sir during an alumni meet at Bapatla. We discussed regarding gene to interaction and predictive breeding. And that interaction actually eventually led to today's lecture. Despite holding highly distinguished international positions, Dr. Prasanna sir graciously accepted our invitation to deliver this lecture. It truly reflects his humility, generosity, encouragement towards our young scientific communities like Share Genes. On the behalf of the Share Genes community, I sincerely thank Dr. B.M. Prasanna sir for this highly insightful and inspiring lecture. We are truly grateful for your valuable time, guidance, and interaction with the participants, sir. I also like to thank the Share Genes, which is formed by a group of 20 young breeders or members who are actually made contributions, voluntary contribution to get Zoom subscription for this particular lecture. And I also thank all the participants for joining and making this session successful. I thank everyone, and I thank Prasanna sir, from bottom of my heart. Thank you, sir.
Thank you so much, friends. It was great to see all my earlier colleagues as well as a lot of young scientists and students participating in this, and enjoyed interacting with you. So my email ID is available. You can, I will type it out. It's b.m.prasanna@cgir.org, or bmprasanna@gmail.com. So if you have any query related to the today talk and what you would like to do, please do send me an email. Okay.
Thank you, sir. Yes,
Okay. All the best,
sure.
and thank you so much. Bye bye.
Yes.